Welcome to the fourth lecture of Bio 120. Today we're going to be looking at how prokaryote cells move and what structures they use for motility. Well, motility. It's a fundamental property of many cells. They need to be able to move in order to get to food, to avoid predators, to colonize different areas. Now, multiple ways of motility exist. The first one is going to be passive motility. Those are the organisms that are carried by air currents or the ocean currents, or microorganisms that are aerosolized. For example, when a person coughs and sneezes, look at the way that this person is coughing and is not covering their mouth, and all those small particles that you can see are particles that potentially could carry microorganisms to the next person. The other way in which microorganisms move is through active motility, in which they actually have structures in their cells that allow them to move within their environment. So why do they move? They move because they want to find nutrients and food. They want to avoid hostile environments. Many microorganisms which are pathogenic move in order to be able to invade the host or disseminate within the host or disseminate across different people, so from one host to the next. And the cause is always the cause of energy. Now, we're going to look at the flagellar motility in bacteria and archaea. So flagella are structures, as we discussed earlier in class, that allow microorganisms to move. And different bacteria have different arrangements about flagella. Bacteria that have only one flagellum are considered to be monotricious, and those are going to be having a flagellum that is going to be in the side of their body. Amphitricious bacteria have two flagellas which are an opposite side of the cell. One, for example, in this image is shown here in the left, and another one is shown here in the right. Lophotricious bacteria has what is called the tough flagellation, which is multiple flagella but all of them coming out of the same side. And peritricious bacteria, like E. coli, have flagella distributed all around their bodies. So here in figure 2.48, what we have in figure A is a peritricious bacteria. In part B, we have a monotricious bacteria, and in part C, we have a lophotricious bacteria. Now, here I'm going to be showing you a set of movies that are going to give you samples of how some organisms move. So we have here Spirillum volutans. It's a microorganism that has an amphitricious arrangement of flagellum. So two polar flagellum that are placed in opposite direction. What we're going to see is a movie about how they move. So let's start the movie again. So what happens is that the flagellum that is located in the direction of movement, it's wrapping itself around the body of the bacteria, while the flagellum that is in the opposite side of the movement direction is the one used to propel the microorganism forward. As you see, that results in the flagellum moving a little bit in a screw cork arrangement. I'm going to stop that movie and go to the next slide. Now, peritricious bacteria, like E. coli, they have flagellum arranged all around their cell bodies. So when they move, what happens is that the flagellum form a bundle in the opposite direction of movement and is used to propel the microorganism. What we're going to see in this movie is an example of E. coli, which is an example of peritricious bacteria that are arranging the entire flagellum structures in a bundle and using them to propel. Notice how the bacteria here are arranging the many different flagellum that they have, and they themselves help propel the organism in one direction. Here's another one coming up from the top. And you can see that the flagellum is much larger than the microorganism itself, and all of them are moving simultaneously, and the flagellum then has, as shown here in the still moving and the still picture, an undulating shape, which allows the microorganism to move. So now, other microorganisms possess flagella, but those flagella are internal, and that is what we call an endoflagellum. Here is an example of spirochetes, which have the flagella inside their body. It is anchored at one pole, and it extends about 2/3 of the body of the microorganism. So because of that, the bacteria moves in a corkscrew movement. So here in the movie to the right, we're going to see some spirochetes as they are moving. First, we're going to see them in bright field microscopy, and actually this is dark field microscopy, and then we're going to see them in fluorescence microscopy. Notice how their movement, as is being followed by the camera, it's showing them moving in a corkscrew fashion. In a moment, the scientist who took this movie is going to change, there you go, it changed the optics to see fluorescence, and they have marked the flagellum molecule, and now they can see the bacteria as it is moving, and there they stop. And you can see the arrangement inside the microorganism where the endoflagellum is present, allowing them to move. The flagellum is one of the ways in which bacteria are able to move, and they're used to propel themselves in a liquid environment. So one of the questions that scientists had is like, how is the motility by flagella driven? And there were two different models proposed at the time. One of them was the model of the whip movement, which it was indicated that the flagellum will be moving up and down like a whip, creating the undulatory waves that we saw previously in that E. coli movie. Other people had the idea that the flagellum will move in a circular fashion, a propeller model, and that circular fashion will create the wave movement that the flagellum was taking, allowing microorganisms to move. So two scientists, Silverman and Simon, in 1973, found a microorganism mutant called polyhook, and that polyhook, instead of having the long flagella of E. coli, had very tiny stubby flagella. They were a defect in the way that the flagella was made, and therefore they're very, very small. So what they did was that they were able to adhere the bacteria into a glass slide, and the bacteria was fluorescently labeled, and they decided to see how the bacteria will move. If the bacteria was moving by the whip model, the microorganism will be moving left to right, as it was attached directly to a solid stratum. But if the bacteria was moving by a propeller motion, the cell of the body will move in a circular fashion. So here is the next movie is going to be the result of that experiment. So what we have here are flagella, where a single flagellar filament has been attached to the substratum, and this is from the Silverman and Simon paper. And what you have here are fluorescent microscopy pictures of the bacteria. As you notice, they're very pixelated. But what I want you to look out is the way in which the cell body of the bacteria move. So here I'm going to start the movie for you. I want you to observe how the microorganisms are moving. There we go. As you see, the bacteria are moving in a circular fashion, supporting the idea that the microorganism is moving in a propeller-like function instead of left to right, as it would be predicted if the microorganisms were moving in a whip-like motion. So the experiment of Silverman and Simon showed then that the microorganism, the bacteria flagella, moves in a circular fashion, and then that led to the question, how it's that movement in a circular fashion accomplished. So what happened was that they then decided to do molecular studies and elucidate the structure of the prokaryotic flagellum. So the prokaryotic flagella is a very complex external structure in microorganisms, in bacteria and archaea, very similar. And it is composed, in the case of a gram-negative microorganism like E. coli, it is composed of multiple rings. Here, the L ring goes across the outer membrane of the E. coli bacteria. The P ring is the ring that goes across the periplasm, and the MS ring is the ring that goes across the plasma membrane, but it also is composed by another ring, the C ring, C for probably being inside the cytoplasmic state. In the rings are connected through a rod shown here in the middle. Now, at the basal part of the flagellum, the area that is part of the cytoplasmic membrane, you have other molecules that are important. You have the MOT proteins, which are motor proteins designed to use as a turbine to design to move the flagellum, and you also have the flea proteins, which are the motor switches, and the motor switches allow the flagellum to change direction. So the flagellum could turn counterclockwise, or when the flea proteins are engaged, it will then change the rotation to clockwise. So the combination of the P ring and the MS and C ring is called the basal body. To the L ring, you have now an attachment of a structure called the hook. The hook gives the flagellum an initial curve, so the flagellum is not sticking out of the outer membrane completely straight. Now, the flagellum part itself is composed of a very large filament. That filament is about 14 nanometers in diameter, but it could be several micrometers in length. Usually, a bacterial cell is about 1 to 2 micrometers in length, and as you saw from the picture with E. coli, the flagellum was about 6 to 7 times longer. So there have been flagellums that have been measured to about 10 microns, very long flagella. Now, the flagellum is composed of monomers of the protein flagellin. We're going to look about that in a moment more carefully. This other picture here, it's a transmitting electron micrograph of the bacterial flagellum showing the L ring, the P ring, the MS ring, and the area where you can have the C ring as well, where the flea proteins and the mud proteins are organized together as a turbine. As you can see, all of that is connected through a rod until it gets to the external part here, which is the hook. So how the flagellar is being made? The flagellum is being made from inside of the cell, and you have about 50 different genes that are required for the assembly and the regulation of flagellum. As you may imagine, you have regulation of location in the case of the polar flagellar or the amphitrachous flagella, so they can be only present in the poles, and you have regulation about the number of flagellum present in the microorganism. So the flagella will start by initiating and inserting proteins from the MS and the C ring in the cytoplasmic membrane. That is followed by the construction of the rod. As the rod continues, it already touches the peptidoglycan layer. Now you have addition of the MOT and flea proteins in the basal body, and eventually you get the construction of the P ring. Once the P ring is constructed, the rod is extended into the outer membrane of the cell wall of E. coli, and there you have the assembly of the L ring. As I mentioned earlier, the rod is hollow, and that allows proteins which are made in the cytoplasm to be transported across and assembled at the location where they need to be. And that is how the hook becomes constructed. Hook proteins are going to be transported through the rod of the flagellum and assembled by their structure into the early hook. After that, you will have the addition of a cap protein, and the cap protein is required for the assembly of the flagellin monomers. As the flagellin monomers now go through the flagellum and into the hook, the filament becomes to grow, and the filament becomes polymerized as more flagellin molecules are being added, and at the very end, the cap molecule is present and helps the assembly of the flagellin monomers into the structure that the flagellum is. So you get thousands of flagellin monomers, being added as the filament grows. I have a movie that I'm going to upload later for you so you can see what is the idea of how the flagellar structure is made from inside the E. coli bacteria. Now, How is it turned around? So the flagellum is one of the most interesting engines in the living world. What we consider is that you have the MS ring and the C ring, and they have the amino acids which are present in the MS and C ring. They're alternating charges from positive to negative. So you may have glutamic acids here giving negative charges, and you can have lysines in the other side giving positive charges. And they are arranged in such a way that they alternate. The way in which the flagellum turbine is powered is through the proton motile force, and it takes about 8,000 protons to do one entire turn of the flagellum. The protons are believed to go through the mock proteins, and as they are passing, they're going to attract some of the negative charges, but when the negative charges pass, their positive charges are going to repel them. So that's going to be then the force that is driving this interchange of positive and negative charges that is attracted and repelled by the protons going through the moth protein are going to cause the flagellum to turn. Now, as the flagellum is turning, about 1,000 protons are being passed. It's believed that in E. coli, the forward motion of the bacteria is caused by a counterclockwise rotation of the flagellum. So in this example of movement, we have a bacteria that has a polar flagellum, so it only has one flagellum at the end of the body of the microorganism, and it is rotating in a counterclockwise position. So some bacteria are able to rotate it in the counterclockwise position that is going to allow the microorganism to move here to the right. And then when they want to change direction, they're able to now to switch the direction of the rotation of the flagellum. And now it's rotating clockwise. And therefore now the bacteria is able now to follow the flagellum as the flagellum is directing the microorganism. So it can move now to the left. So the microorganism is able to move from right to left by changing the direction of flagellar movement. And that is happening if the flagella can reverse direction. But not all flagellums are able to reverse direction. So microorganisms have what is called the unidirectional flagella. So in this case, the challenge is how do you change direction? So the microorganism, for example, will be moving its flagellum counterclockwise, clockwise, so the microorganism is moving, then it stops flagellar movement. What that is going to do is to allow the microorganism to move by Brownian motion. So it's relocating the microorganism randomly. And then, at a certain point, by whatever cues the microorganism has, it's going to activate flagellar movement one more time. And now is able to move into a different direction. So these are the two ways in which microorganisms with polar flagellum are able to move. Either because they're able to change the rotation of their flagellum from counterclockwise to clockwise, or because they stop, allow the Brownian motion to reposition them, and then engage the flagellum one more time. So here, it's an example of Rhodobacter spheroides. This is an organism that has only one flagella. It's a polar flagellum, and it is emitted from their body. And what you're going to see is that the filament is going to spin, usually clockwise, and form a helical structure. These are going to be very speedy microorganisms, so let's just watch them in the movie. And as you can see, they are just zooming by, and here they come back into focus, and they're going around. This one over here in the middle stopped, and is reorienting itself. And eventually, if the movie would have been longer, the microorganism would engage again the direction and start over. So let's just play that one more time. So some of them are just turning around. All of a sudden, this one over here stops. It stops again and reorients. Now, that poses a challenge for bacteria who have a peritricious arrangement, because as the bacteria begin to engage their flagellum for movement, their flagella all bundle into one side, like a ponytail, and how then does it change direction? So what it was observed is that flagella that have the peritricious arrangement they have a change of pattern. One of them which is going to be the run, and another one which is going to be the tumble. So the bacteria will begin moving. Here is the bacteria going like E. coli to the right. It's going to bundle the flagella. The flagella is going to be moving in the counterclockwise direction. But at a certain point, by mechanical reasons that we're going to discuss later when we talk about chemotaxis in the molecular sense, the flagella stop and they start to rotate in the opposite direction. That means that the bundle of flagella falls apart and they all now distribute everywhere. That makes the bacteria move and change direction. And eventually, when the bacteria the flagellum, are engaged again in the counterclockwise rotation, they begin to move again. So these microorganisms are moving by activating run and tumble phases. So the run phases allow the microorganism to move, the tumble phase allows them to change direction. Now, during movement, what you have is a random event where the tumble and run phases are alternated, and the bacteria basically doesn't move anywhere in particular. They're just moving in their environment, and it doesn't go anywhere. So it will move, it will have a run phase, it will tumble, the bacteria have another run phase, it will tumble, change direction. Now it will then have another run phase, tumble, change direction, run, tumble, change direction, and continue moving. So it could have a run phase for a second, have a counterclockwise for about 0.1 seconds, and that will change the direction of microorganism, and then they will start another run phase. But we're going to take a look at that right now. So what I want you to see, this is our E. coli bacteria. E. coli are going to be having the run and tumble phase. So I want you to observe how the microorganisms are doing that. As you see, they're very quick. And this one over there in the middle, and it went out of the phase. I'm going to play this one more time. The microorganisms are moving, they tumble, they change direction because their flagella goes all over the place into the tumble, and then they change. One more time for you to see. So they have a run phase, they have a tumble phase, and they initiate another run. So that basically leads for the distribution of bacteria in a solution. So now, let's think about this for a moment. So we have about 50 genes involved in flagellar motion. They can propel the bacteria quite fast, about 60 cell lengths per second. If bacteria is about a micron, you can think that it's moving 60 microns per second. The movement of the flagellum, it's propeller like in mechanism, so it's going around in a circle, and it depends on the flagellar arrangement. So bacteria that have a polar flagellar are going to move different than bacteria that have a peritertious arrangement. Now, energy wise, it requires the proton motile force. In E. coli, about 1,000 protons per rotation, and E. coli moves around 270 revolutions per second. So that flagellum, it's pumping quite fast. So if you multiply 270 revolutions per 1,000 revolutions per 1,000 protons per rotation, you get that you require 270,000 protons to move per second. Now, Vibrio alginolyticus, which is a different kind of microorganism, also gram-negative, it has a much faster flagellar spinning. It's about 1,100 revolutions per second. So think about the energy cost in protons that flagellar movement requires for microorganisms to have. Now, when we compare the speed of a bacteria, they are pretty fast. When we think about them when we think about them as cell lengths per second. So, as I mentioned, an E. coli can move about 60 cell lengths per second, and that is 0.00017 kilometers per hour. So, by our standards, that is pretty slow. The cheetah, which is the fastest mammal on the planet, it's moving 110 kilometers per second. But when you consider body length, a cheetah moves about 25 body lengths. So less than half the speed of E. coli when we think about it. At the time that I made this lecture, Michael Johnson, which is an Olympic gold medalist, now I think I have to go for Usain Bolt for this, was having 10.3 meters per second was his speed. And he was moving about 5 body lengths per second, so even slower than the cheetah, but and much, much slower than E. coli. So when we consider the unit cell lengths per second, bacteria move a lot faster than mammals do. So it's actually very fast, and it allows them to get to where they need to go. So now, part of the idea about movement that we had is that bacteria respond to cues in their environment, and we call this taxis. So taxis is the idea that you're going to have movement to or away from a signal. And many different signals could be the ones that make microorganisms move to them. You have, for example, phototaxis, bacteria that move towards light. A lot of the photosynthetic microorganisms are going to move towards the bright light source. Some of them are going to have thermotaxis, which is heat. Bacteria which require oxygen are going to show areotaxis. So those of you taking the lab are going to see that Pseudomonas, the bacteria that we're going to be isolating in lab number 3, have areotactic behavior because they are respirers and non-fermenters. So they always have to go for where the oxygen are. We are areotactic as well. We don't want to be in an environment without oxygen, otherwise we choke. Osmotaxis, it's going to allow microorganisms to move to a particular osmotic strength. And an interesting kind of taxis is magnetotaxics. They are bacteria that has internal structures that allow them to move to magnetic fields. And of course, the kind of taxis that you have studied a lot is chemotaxis. So now, in this image, what we have is a sample of how we study taxis. We have in figure a microorganism colony in liquid, and we can put a capillary tube which has the same liquid where the microorganisms are growing. And what we then do with time is to assess how many microorganisms have entered the capillary tube when we either put a control solution or an attractant like a food source, or a repellent like a poison that may kill the bacteria. So what we see here in B, it's the experiment showing taxis for microorganisms against, for example, glucose. And as you can see with time, the number of microorganisms inside the capillary tube have gone up. In the control, the number of microorganisms, they go in, but it doesn't change compared to the outside. And when you have a repellent, the number of organisms inside, it's very low. So this can be graphed now by mapping, this can be seen over here, by graphing the number of bacteria inside the tube at different time points. As you can see, with time, in the control, the number of microorganisms are going to be moving in through random motion, are going to go up, but at a certain point it reaches a plateau because no other microorganisms are going to be going in and out. There is no reason for them to be attractive to the control. In the case of the sugar, like an attractant, the number of organisms increases with time until it reaches a plateau because most of them have gotten over there inside the tube. In the case of a repellent, the number of organisms remains constant throughout the entire experiment, indicating that no organisms are going inside the tube, but that they're actually going away from it. So this was the initial idea that microorganisms have a way to sense their environment and therefore react to it by moving towards different sources of either attractant, repellent, or one kind of source or another. Now, what are required for microorganisms to move? They need to be able to sense the external signal. So that means that they have a mechanism to detect that outside of their system the signal is present. They need to now transmit that signal intracellularly. Remember, most of the receptors are going to be in the plasma membrane, and like we study in Bio 110, now that signal has to be transmitted inside, so therefore that signal becomes a response. So once we start to look more carefully about the molecular mechanisms of this, we're going to look and see how the microorganisms respond to their environment really efficiently. So the other thing that is important for this is that the system has to be scalable. It needs to adapt to the intensity of the signal. So a microorganism moving to a source of food will be able to tell that the food in concentration is increasing. Now, here is when the tumble and run movement becomes important for bacteria. In this image, you have a concentration of an attractant shown here in green, and by diffusion, the attractant becomes more diluted and more diluted as you move away from the source of the attractant. Now, as the bacteria is moving in its environment and is beginning to sense that there's some kind of attractant present, the microorganism is able to make temporal comparisons. So it's going to compare the concentration of attractant at one point and later at a different time. It responds then by comparing the amount of signal, the level of intensity of the signal at the different times. And therefore, it's able to decide how and where to move. The frequency, the response to that sensory, it's the change in the run frequency. So the microorganisms then become and have longer runs and fewer tumbles. So once the run stops and it tumbles, it's able to say, well, you know what? I am detecting less attractant this way, therefore I must be moving away from the source. And it is then measuring the amount of attractant with time, and eventually all the microorganisms land where the attractant concentration is the highest. So this chemotaxis in bacteria is a type of sensory response system in the same way that we have it in neurons, analogous to it. Now, flagella, it's not the only way in which bacteria can move. We're going to see that you have twitching movement, which is based on the pilus. You have gliding motility and slime motility. So let's take a look at those interesting ways in which bacteria move without a flagellum. Now, as we mentioned earlier, bacteria have extensions called the pilus. And what we see over here in this electron micrograph, it's bacteria extending very long protrusions. These are not their flagellum. These are their pilus and their fimbriae. And what happens is that when the pilus is extended, the pilus can bind to the substrate, and eventually the bacteria retract the pilus. But because the pilus is anchored to the ground, the bacteria body then gets dragged towards the direction of the attachment. And we call that the grabbing hook model. What we're going to see here are Pseudomonas aeruginosa, which also display this kind of movement. As you know, as I mentioned to you earlier, Pseudomonas have a pilus, and this is what is called a type 4 pilus, which is about 6 nanometers in diameter. And what we're going to see from the movie in here is how the pilus can be extended. Note that now, in this case, the pili are being retracted inside the microorganism. So they're becoming smaller. Now, if the microorganism were to be attached to a stratum, what is going to happen is that the body gets moved. So now let's look at the second movie showing this phenomenon. It's very fast. Let me do it one more time. As you see, once the pilus is attached, the microorganism can drag itself to the position of the pili. So this is a mechanism in which bacteria like Pseudomonas, Mycococcus xanthus are able then to use their pilus to move. And you must imagine, if you create mutations which lack the pilus, or the pilus that cannot be retractive, those microorganisms lose their capacity to move. Therefore, the retraction powers the twitching movement to allow the microorganism to drag itself across. Another way in which bacteria move is by gliding. So this movement lacks flagella and lacks pili. So the microorganisms are gliding nicely over the surface. Whereas flagellar movement requires to have a liquid media, so they're swimming, pili and gliding motility require to have a substrate, something for them to attach themselves on. So what we're going to show you here, these are mycoplasma, and as you can see, they are gliding beautifully over the surface of the slide. They're not really going up and down like the E. coli were doing, coming in and out of the focus plane because they're swimming in 3 dimensions. Now these microorganisms are moving in 2 dimensions. So this kind of media is sufficiently viscous for us to slow down the movement of the bacteria. So there's ideas of how this is happening, and there's 2 models. One of them is the racetrack model, another one is the slime extrusion model. Now, in the racetrack model, here you have a gram-negative microorganism. They have proteins that are going to allow them to attach themselves to the surface. And through mechanisms of movement of protons, proteins that are in the plasma membrane are going to power the ricocheting of the proteins at the surface. So if the protein is attached to the surface, that protein is going to be then track and move, sort of like the way in which you move the track of a tank backwards. And as you see the movement of the outer gliding proteins, the ratcheting is making that protein move towards the tail of the organism, which causes the entire organism to move in the opposite direction. Flavobacterium johnsoni is one of the examples of bacteria that uses the racetrack modeling system. And I've been trying to see if I can find a movie to illustrate this better, but so far, unfortunately, I haven't been able to do this. So I apologize to you for that. The other type of gliding motility, and I think this is pretty darn awesome, it's the ejection of slime. And cyanobacteria are bacteria that have this kind of gliding motility. So the bacteria have channels called slime nozzles that are present in the rear of the microorganism. And what they do is through helical filament bundles that allow them to contract, they are then able to extrude slime out of the rear of the microorganism through the slime nozzles. The slime attaches to the stratum and helps the bacteria move in the opposite direction. So what I'm going to show you here is a movie of cyanobacteria gliding, and you can see how smoothly the bacteria glide. Before actually I show you the movie, let me show you this scanning electron microscopy image of the butt end of a bacteria, and you can see the trails of the slime being ejected outside as the bacteria is moving. So the slime, part of the interesting part about it is that it's somewhat dehydrated inside the bacteria, but when it is extruded out, it becomes hydrated and expands, and that helps push the microorganism. So let's just watch this movie. Let's see how this is going to do. Over here, I think I have to do this now. There we go. What you can see now here is cyanobacteria. This is the body of the microorganism. You can see how nicely it is gliding. It's a slow kind of motility, but it allowed the microorganism to move towards, in this case, cyanobacteria, towards light. I don't know if you can appreciate at the end of the microorganism how the slime made a little bubble over there and the bubble stayed behind. But you can see that it's slowly being generated and pushing from the rear the microorganism in a beautifully gliding motion. Here we are. Okay. So I put the address of the movie here for you to watch at your leisure when you want and have time. Now, let me show you another kind of gliding bacteria. And these ones are moving, there's our Xenococcus, and they're moving very fast. It moves about 25 microns per second. So it's a very speedy bacteria that move without flagellum. And they have what is called this pinwheel rather than tether cell motion. So here we go. As you can see, the microorganisms are zooming in only 2 dimensions. So you see them just go around the dimensions of the slide. Some of them can go a little bit up, some of them can go a little bit down, but they are moving very quickly. They have these little hairs in the body, which are about 0.15 microns long. And the hairs, they go through the outer layers of the cell wall into the membrane, and the energy of motility is the electrochemical gradient that allows the microorganisms to swim like if they're pushing the little hairs. So that allows them to glide very graciously in their media. So let's take another look at last group of microorganisms. Here we go. I want to show you how Myxococcus santus, it's a bacteria that move. They display what is called cooperative gliding. And what they're going to see in this video that I'm going to show you is pretty remarkable. I'd really like to see this. How they actually go and hunt a neighboring E. coli colony. So what the slide is going to show you, it's the Myxococcus santus, is a microorganism that displays cooperative gliding, which means that they're going to move as a unit. They coordinate their movement, in this case, to hunt fellow E. coli bacteria. And what we're going to see is that as they are attacking the E. coli colony, they change their movement to something called the rippling movement. Once they have destroyed all the E. coli and eaten them, and they ran out of food, they're going to activate a developmental program similar to sporulation, in which they're going to become fruiting bodies. And that's also going to be shown here. So here is the address of the movie, but I'm going to show you the video right now. It doesn't have any sound, but it has little tidbits of information that come up. So here we are. As you see over here, the Myxococcus is expanding. And now they have to attack the E. coli, and it is lysing the E. coli cells. Notice that the killing of the E. coli is only happening at the contact of the bacteria. And you have this oscillation movement, which is called the rippling. As they go and destroy all the E. coli, the bacteria begins to starve because they don't have enough food. And that's once they start activating the alarm system similar to sporulation, where they become fruiting bodies. As you can see, that the ones that are left behind where there's no more food, they begin to form the fruiting bodies. As the movie said, the last of the rippling bacteria over here, once the food is completely gone, they activate the program to become fruiting bodies. This is very cool. All right. And with that, let me go back to the lecture. There we go. With that, we're going to end our lecture. What I'm going to show you, it's that all these videos were generated at the Roland Institute at Harvard University, so you can go to their website and actually see the index of movies. Have a good weekend, and I'll see you on Tuesday.